lecture 2, that is models of embryology. So this is the next model of primitive gastrointestinal tract, 25 days of embryo. Now you can see here what are the structures visible in this model. The stomach is visible, then liver bud is visible, then duodenum, midgut, hindgut, cloacal membrane is there, then allantois, vitelline duct and heart. In this, the larynx, then esophagus and trachea, that lung bud is there, so that is visible. Then stomach, duodenum, pancreas is here, then primary intestinal lobe, cloaca, liver and septum transversum. So this is the model of primitive gastrointestinal tract. Now what are the commonly asked question in Viva is what is septum transversum? The answer is it is thick plate of mesodermal tissue occupying the space between thoracic cavity and stalk of the yolk sac. What are the embryonic structure forming the liver? That is endodermal bud, umbilical and vitelline veins and septum transversum. Now endodermal bud is arising at the junctional point of foregut and midgut, but on the ventral side. Then umbilical and vitelline veins, they are lying in septum transversum. Now name the structures arising from septum transversum. The answer is capsule and fibrous tissue basis of liver. Then what is the fate of allantois? It is median umbilical ligament of urinary bladder. Now this is the next model of relationship of the hindgut and cloaca at the end of 5th week. Now what are the structures visible? You can see here mesonephric tissue is there, then mesonephric duct is there. Then here the ureteric bud and metanephric blastema. Now cloaca is visible in this model, urorectal septum is visible, allantois is visible, and this is hindgut. Now this ureteric bud penetrates the metanephric blastema. Now what is the main important question in this model is what are the subdivisions of cloaca? So urorectal septum divides the cloaca into two parts, that is primitive urogenital sinus and rectum. So rest things you can understand that the development of the kidney can be asked. So what are the derivatives of the ureteric bud? So these things can be asked. Coming to the next model showing the development of the stomach, you can see the tubular form, then relative growth of the left border, then lesser curvature and greater curvature is formed. Pylorus is now visible in this. Then this is the fully developed stomach showing the greater curvature, lesser curvature, pylorus, and esophagus is continuing at this portion, that is cardiac end of the stomach. Now keeping in mind this model, you should know the different derivatives of the foregut, that is very important. So midgut derivatives can be asked, then hindgut derivatives can be asked. Coming to the next model, that is the development of the pancreas. So this model, first part is showing the trachea, here it is the larynx, then this is lung bud, then esophagus, stomach, dorsal pancreatic bud, ventral pancreatic bud is visible, gallbladder is visible, and liver bud is visible. Now here you can see some with rotation of the stomach, the dorsal pancreas, ventral pancreas, gallbladder, stomach and esophagus. Now here the better defined structure of the dorsal and ventral pancreas. Then rotation, with the rotation of the gut they come on this side and the gallbladder is coming on this side. So here cystic duct is developed, then common bile duct is developed. So these are visible in this model. So keeping in mind what are the questions which can be asked is, so what are the sources of the development of pancreas? So two endodermal buds at the junction of foregut and midgut, dorsal and ventral buds. Which part of the pancreas is arising from dorsal bud? That is upper part of head, neck, body and tail. Ventral bud is forming lower part of the head and unseen process. So what are the pancreatic ducts? Main pancreatic duct, which is also called as duct of Wirsung, which is opening into the second part of the duodenum at the major duodenal papilla, and accessory pancreatic duct or duct of Santorini opening into minor duodenal papilla. So what are the different anomalies of pancreas? So these are the anomalies that you should know. So out of this, the annular pancreas is very important. What is that? The ventral pancreatic duct bifurcates and surrounds the second part of the duodenum like a collar, causes the obstruction of the duodenum. Now this is the model showing the successive stages in the development of the trachea and lung. You can see the tracheoesophageal ridge is visible, foregut is visible, and respiratory diverticulum is there. So this respiratory diverticulum is dividing into these lung buds. Now later on the subdivisions of the lung bud is happening in this, and here the development of the lung is shown. In this right and left bronchus is visible. So on the left side upper and lower lobe, and the right side right, middle and lower lobe is visible. So what are the questions which can be asked keeping in mind this model? What is tracheoesophageal fistula? You know it, there is the communication over here. Sometimes they are rising from the same structure, so communication is there. So this is the abnormal communication between trachea and esophagus. So what is the number of total bronchopulmonary segment? 10 on each side. What are the stages of lung development? Pseudoglandular period, canalicular period, terminal sac period, and alveolar period. So these are the stages of lung development. Theory you can study from your textbook. Now this is the next model showing the development of the urogenital sinus into urinary bladder. So in this model what is visible? So this is the urogenital sinus, the two parts, that is definitive urogenital sinus and pelvic part of urogenital sinus. Then here the ureter is there and allantois is there. Now from this portion the ureter is arising, here the seminal vesicles, then this is anorectal canal. In this model, so further growth is happening. So prostatic and membranous urethra is formed, the penile urethra is visible, seminal vesicle, ductus deferens and prostate gland you can see here. So this is now urachus because this allantois is obliterated. Now in this model the development of the urogenital sinus into the urinary bladder and the definitive urogenital sinus. Now in the male this definitive urogenital sinus is forming the penile urethra, the prostate gland is formed by buds from the urethra, and seminal vesicles are formed by the budding from the ductus deferens you can see here. So seminal vesicle is formed here and prostate gland is formed by the buds from the urethra. Now this is the model showing the ascent of the kidney. You can see in this what is shown, that is tubules are shown, mesonephric tubule is visible, then yolk sac, allantois and cloaca is visible. So further growth is happening. So this metanephric duct is visible initially. So now here the gonad they appear, mesonephric duct, kidney is appearing here, allantois, urogenital sinus and ureter. Now further, because it is arising in the lower portion, it has to ascend. So the ascent of the kidney is there. This is the permanent kidney. Then testes, epididymis, urethra and phallus is visible. So these models are showing the successive stages in the ascent of kidney. So definitive human kidney is derived from the metanephros, which lies in the sacral region. Differential growth of the abdominal wall causes the kidney to ascend to the lumbar region. So as the differential growth of the abdominal wall is happening, it ascends upwards. So this is the third stage from where the progression of the ascent of the kidney is happening. So this is the initial stage, this next stage, and this is the last stage when the permanent kidney is taking its original position in the lumbar region. Now what is the source of origin of human kidney? So each kidney consists of two components, excretory system and collecting system. Collecting system is developing from the ureteric bud. Excretory system is developing from metanephric mesoderm. What are the components of excretory system? Bowman's capsule, PCT, loop of Henle and DCT. What are the components of collecting system? Ureter, renal pelvis, major calyces, minor calyces, collecting duct and collecting tubule. So they are arising from ureteric bud and they are arising from metanephric mesoderm. This model is showing the dorsal view of the bladder, showing the relationship of the ureters and mesonephric duct. So mesonephric ducts are visible. The ureteric bud, these are the yellow colored structures which are visible in this portion. So they are missing in this first part A. Then posterior wall of the urinary bladder, then trigone is visible, then ureter is there. This is the trigone, mesonephric duct is there. So in first model this ureteric bud is basically missing. So here the ureteric bud should be there. So you can see the stump of that portion. Now initially ureters are formed by an outgrowth of the mesonephric duct in this region. Then assume a separate entrance into the urinary bladder. Then trigone of the bladder formed by incorporation of the mesonephric duct. So that is how the stages is happening and how they are related with each other. From which structure ureter is arising? It is arising from the ureteric bud, from mesonephric duct. What is the fate of mesonephric duct in females? These are the three structures, that is ureteric bud, trigone of bladder, posterior wall of the part of female urethra. So this is the fate of mesonephric duct in males. So these are the structures which are arising from this that you should know. So what is the fate of mesonephric duct in females and what is the fate of mesonephric duct in males? Now this is the next model showing the descent of the testis. So you can see here the testis, then caudal genial ligament and gubernaculum. This is phallus, this is rectum, this is symphysis pubis. Now testis, vaginal process. In the second model you can see here the testes are descending downward. So this is the gubernaculum testis, this is the vaginal process, this is the scrotal swelling, this is symphysis. Next, what is happening? It is coming downward. So these are the structure. Then the coverings of the testis, they are formed, that is parietal and visceral layers of tunica vaginalis and gubernaculum testis. So this is during the second week, this is third week, this is 7th month and shortly after birth. Now what are the questions which can be asked? In which region of the abdomen testes start developing? That is lumbar region. Different location of the testis during descent, that is iliac fossa, deep inguinal ring, inguinal canal and scrotum, and it is 3rd month, 7th month, 8th month and 9th month respectively. Factors responsible for the descent of testes. So these are the factors which are responsible for the descent of testes. Coming to the formation of the uterus and vagina, in this model uterine tube is visible, uterine septum is visible, sinus tubercle is visible, urogenital sinus is visible, then caudal tip of paramesonephric duct is visible. So this is caudal tip of paramesonephric duct. Then tissue of the sinovaginal bulb is visible, then lumen of the uterus. Then this is the formation of the uterus with cervix, vagina and fallopian tubes. So at 9 weeks, this is the picture. At the end of 3rd month, this is the picture, and newborn baby has this picture. Now basic highlights of this model is fornices and upper part of the vagina are formed from paramesonephric duct. Lower portion of the vagina is formed from sinovaginal bulb. Then important congenital anomalies of the uterus vagina is visible, that is absence, unicornuate uterus, duplication and absence of fallopian tube, duplication or absence of vagina. Vagina may have abnormal communication with the rectum, then it is called as rectovaginal fistula. Urinary bladder has if communication with the vagina, it is called as vesicovaginal fistula. Coming to the development of eye, in these three models, in first model optic cup is visible, optic stalk is visible and choroid fissure is visible. Now this section is taken from here, at the level of optic stalk. So you can see the outer layer, inner layer, then hyaloid artery is present in the choroid fissure. Now section through the lens vesicle, the optic cup and optic stalk at the plane of choroid fissure if it is taken in this direction. So intraretinal space is visible, wall of brain is there, lens vesicle, hyaloid artery and lumen of optic stalk. So this section is taken along the length of the choroid fissure. Now these important highlights of this model is, so components contributing to the development of the eye, an outgrowth from prosencephalon, then specialized area surface ectoderm, lens placard give rise to lens, prosencephalon is going to form optic cup and mesoderm is contributing to the development of eye. So this is important. Then choroid fissure is, what is the structure, but basically how it is arising. The margins of the double layered cup grow over the upper and lateral side of the lens to enclose it, but this growth does not happen on the inferior aspect and shows deficiency inferiorly, and this deficiency extend along the inferior surface of the optic stalk. We call it as choroid fissure. Formation of the fissure allows the hyaloid artery to reach the inner chamber of the eye. During the 7th month, lips of the choroid fissure fuse and mouth of the optic cup becomes future pupil. Coming to the next model, that is formation of the nephric tubule. In this, the basic structure which are shown in this model is paraxial mesoderm, intermediate mesoderm and somatic mesoderm and subplanchnic mesoderm, or you can see the lateral plate mesoderm. Then coming to the these structures what are arising from this, that is internal glomerulus, external glomerulus and nephric tubules, they are formed. So communication between the endoembryonic cavity and nephric tubule is shown over here. So this model is showing these structures. So important highlights is that urogenital system has two components, urinary system and genital system. Both the systems develop from intermediate mesoderm and excretory duct of both the system enters into the cloaca. So these are the important things regarding this model. Now this model is showing the formation of the atrioventricular valves and chordae tendineae. So thick wall of myocardium is shown, then muscular cords are shown, lumen of ventricle is shown, myocardium is shown, then atrioventricular valve with chordae tendineae. Through this it is attached to the papillary muscles. So these are the papillary muscles. The valves are hollowed out from the ventricular side but remain attached to the ventricular wall by chordae tendineae. So these are the models which we have discussed. So thank you very much and good luck. If you like these two lectures, kindly comment in the comment box. Thank you very much.